Good afternoon, everyone. I am Natalie Piperidis, and I am a cytogeneticist at Sugar Research Australia and based in Mackay. I would like to begin by acknowledging the Aboriginal and Torres Strait Islander people of this nation. We acknowledge the traditional custodians of the lands of which SRA is located and where we conduct our business. We pay our respects to ancestors and elders, past and present. The project is the culmination of 5 years of collaboration between Professor Ben Hayes' teams at University of Queensland and Sugar Research Australia. The research is funded by the Department of Agriculture and Fisheries, Sugar Research Australia, and the University of Queensland. It is my privilege to extend a warm welcome to each of you at the launch of the 2024 SRA webinar series. I am pleased to see such a substantial turnout, anticipating that today's session will be a foundation to the latest advancements in sugarcane research. Our aspiration is for the 2024 series to evolve into an admissible event, providing insights into the dynamic landscape of sugarcane research. Today's webinar will consist of a presentation of around 45 minutes, following by a Q&A session for 15 minutes. So during the presentation, we ask that all microphones are on mute, and then questions can be asked during the Q&A session at the end of the presentation. However, you can also type questions via the chat function at any time. Open this by clicking on the speech bubble icon located on the bar at the top of the screen. To ask a question during the Q&A session, either type it into the chat or raise your hand using the hand buttons, also located at the bar on the top of the screen. Upon completion of today's webinar, it will be uploaded to our website. So now, I would now like to introduce our first presenter for the series, Professor Ben Hayes. I am pleased to share that Professor Hayes eagerly accepted the opportunity to deliver this presentation. And I'm confident that his enthusiasm will resonate with you as he discussed his finding on accelerating the development of new sugarcane varieties with genomics. Professor Ben Hayes served as the Center Director of Genetics and Genomics for Livestock and Crop Breeding at QAFI, the Queensland Alliance for Agriculture and Food Innovation at UQ. With an extensive background in genetic improvement across livestock and crops, Professor Hayes has been at the forefront of integrating genomic information into breeding programs. He has successfully led numerous large-scale projects, contributing significantly to the implementation of genomic technology in both livestock and cropping industries. As a co-inventor of genomic prediction research, Professor Hayes brings unparalleled expertise to our complex sugarcane crop. And today, he will demonstrate how genomic selection can accurately predict the performance of key sugarcane traits. The application of this technology holds great promise for enhancing plant breeding health. I'm sure you will have many questions for Ben, and you will have plenty of time to address them after this presentation. So without further delay, I will now pass the virtual floor to Professor Ben Hayes. Thank you, Ben.
Thank you, Natalie. So I'll just get my presentation up. Okay, thank you so much. And as Natalie said, I'm actually quite excited to be able to describe some of our results in this project, because as I hope you'll agree, I think it's been quite successful. So before I start, I'd just like to say this has been a great collaboration with the breeders and scientists at SRA. So thank you hugely for their input, and I'll acknowledge them again at the end. Okay, so just to point out the obvious, the aim of producing new varieties for growers is so that the growers can grow these varieties with higher yields, higher sugar content, better fibre characteristics, and varieties that are resistant to major diseases to hopefully make their operations more profitable. So the next question is, how do these new varieties get produced? So over time, how do new varieties get developed? So at SRA, and this is the breeding program for Australia, and many other countries or other countries that grow sugarcane around the world have similar types of breeding programs. These breeding programs are designed to really take the best genetics in a pool of old varieties or old parents, mix them together and create out of that new varieties that meet those criteria, like higher yield, higher sugar content, and so on. Just to go through it quickly, in the Australian program, it looks something like this. Parents, which might be really past varieties that have performed really well, are crossed, and you can see one of the crossings up there at Moringa. That's what this photo is of. From that, you get progeny produced, lots of little seedlings. They get assessed. That cross gets assessed in a progeny assessment trial. From that, the most promising seedlings go forward to be assessed in a more rigorous trial, which starts to look at cane yield and so on in a clonal assessment trial. Clones from there that are really promising go on to a much larger trial across a range of different environments. And finally, a few clones from there fall out as really promising new varieties to go out to the growers. So this system is a pretty tried and true system. It results in new varieties every so often. But I have to say the cycle time, or the downside of it, is the cycle time is quite long. It can be 10 years or even longer to recognise, identify those new varieties and get them out to growers. So what we'd really like to do is speed up that cycle time, get out these new varieties, identify them earlier, get them out to growers more quickly, so they can take advantage of higher yields, better disease resistance, and so on. So how are we going to do that? Can we accelerate these development of these new varieties? And genomic technologies is one possibility for doing this. And the genomic technology I'm going to focus on is called genomic selection. And this is a technology that's actually been implemented in lots of different livestock species and crops. So to explain to you how genomic selection works and how it picks out the individuals with better genetics, the starting point for genomic selection, what you need is a reference population. You need a really large number of individuals with the traits you're interested in recorded on. So in the case of sugarcane, it'd be clones with yield information, sugar content information, disease information, and so on. You need those individuals recorded. The other thing you need is the ability to peer in their genomes and look at what genes they're actually carrying. So you need what we call marker genotypes. And the trick with genomic selection, or the great realization we came to several decades ago, in fact, is that for many of the traits we're interested in, it's not just going to be one gene driving these traits, it's multiple genes across the genome. So for example, for cane yield, it's very unlikely, and in fact it's not the case, that one gene just drives increased cane yield. It's much more likely from all the research that's been done that many different genes across the genome drive cane yield. So what you actually need is genome information or DNA marker information right across the genome. to track all those genes that contribute to a trait like cane yield. That's the case in sugarcane. I've also worked a lot in dairy cattle. A trait like milk yield in dairy cattle is also driven by lots of genes. So to make this thing work in dairy cattle, we also need lots of markers across the genome. And it's the same in any other agricultural species. Once you have lots of DNA marker genotypes or information across the genome, what you can do is line it up with all your trait records for your individuals to start to work out which DNA markers are actually linked or associated with your trait, and start capturing those in what we call a prediction equation. So, for example, at a particular marker, one lot of individuals may carry one variant, say it's A, another lot of individuals may carry T, and if we see a systematic difference, a difference in their performance, so for example, all the clones that have the A allele perform really well, have high yield, all the clones that have a low yield have the T, we know that marker is associated with yield. But as I just said, the likely outcome is actually that we'll have just a relatively small difference at a particular point on the genome, and we'll need to add up across lots of points on the genome to capture all the genes that contribute to the yield. But once we know that information, or the computer's sorted it out for us, we're in a really good position, because what it means is we can go to any individual, say it's a seedling, extract the DNA from that seedling, and based on its DNA information, its marker genotypes, we can actually predict what its performance is going to be, what its yield is going to be if it's grown out in a paddock, for example, in a field. This technology, genomic selection, has been very widely adopted in livestock industries around the world, so dairy, beef, sheep. And in fact, in the dairy industry, it was implemented about 15 years ago now, and we've got really good evidence that it's actually doubled the rate of gain for traits like milk production. So it's led to this doubling of the increase in genetic gain for dairy cattle. It's also been quite widely implemented in crops, and a really nice example is what's happened in the US, and a company called Pioneer used it to identify maize hybrids that were more drought tolerant. So they did that over a period of time, and they released these new varieties called Aquamax that were more drought tolerant. They were able to produce the same amount of corn with less water, and they're now widely grown across the US. The technology's also being used in sorghum here in Australia, and a few years ago we actually applied it to macadamia nuts as well. So it's been quite widely applied across many different crops and livestock species. But let's focus again in on sugarcane. So what do you need to make it work in sugarcane, or what are some of the challenges in sugarcane? The first thing you need is some way of getting this DNA information relatively cheaply on a large number of varieties or clones of sugarcane, so you can set up your reference population. One way of doing this is through a technology called a marker array. And you can see a picture of one of these things here. And we were very fortunate at the start of this project that SRA and CSIRO had just done some really hard work to make one of these marker arrays, which interrogated 50,000 points across the sugarcane genome on one of these arrays in a cost-effective manner. So we could do thousands of clones, screen thousands of clones for these markers right across the sugarcane genome. So we were very lucky that that was developed in a previous project. Another interesting point to consider is most of the species we've worked with in the past, where we've implemented genomic selection, their genetics is actually quite simple. They inherit one chromosome from their mother and one from their father. So they have two chromosomes, just like humans do. They're diploid. Sugarcane, for better or for worse, is much, much more complicated than that. Sugarcane that is grown today is actually the cross of two different species. So it's a hybrid. So it's got genomes coming in from those two different species. And in one of those species, the genome is copied over and over and over again. So instead of having just two copies of each chromosome, like we do, one from mum and one from dad, a sugarcane plant can have anywhere from 8 to 12 or even more copies of its chromosome. That's probably the reason sugarcane grows so tall, is because it's got all that DNA driving increased growth, but it also makes the genetics quite complex. Some people said at the start of this project back in 2016, oh, you're never going to make genomic selection work in sugarcane. The genome's just too complex. We said, oh, we recognise that, but we're going to try it anyway. So we have. So to get genomic selection working, the first thing you need is this reference population of, in this case, clones, where you have performance, yield, cane sugar, and so on, really well characterised. And you also have these snapshots of the DNA across the genome, these marker genotypes. So you need to build your reference population. So through a lot of hard work with SRA and people at UQ, we built this reference population over some time. And what we did was take the final assessment trials, the clones grown in the final assessment trials from 2013 to 2017, and we genotyped them for this SNP array. So we had their final assessment information for yield, cane sugar and fibre, and we also had 50,000 snapshots across the genome for those clones. And in this table, you can just see where those clones actually came from. So we had 338 from the northern region, 230 from the southern region, and these numbers, like northern and southern, they're the number of clones that overlap across the region. But you can see we've got a pretty fair distribution across the final assessment trials grown in the different region. The next step we did was to see how good we were at predicting how the clones performed in that final assessment trial in 2017. So the last final assessment trial. What we actually did was take all the clonal performance from 2013 to 2016. We took their yield information, we took their DNA marker information, we came up with a prediction what each individual marker would do using that reference population, 2013 to 2016, and then we predicted performance in 2017. So just using the DNA marker information on the clones in 2017, we predicted how well they would yield, what their commercial cane sugar would be, and what their fibre would be. And then to see how well our genomic predictions performed, we correlated that with what they actually did, their real performance data in 2017. And this is the answer. So this is the correlation of what we predicted and what actually happened in 2017. So for total cane harvested, it was 0.3, commercial cane sugar, it was 0.47, and for fibre, it was 0.5. So what does that actually mean? It means we're getting moderate accuracies of prediction. To unpack those numbers, to show you what those numbers actually mean, if you got an accuracy of prediction of 0, it would mean you can't predict anything at all. You're making a DNA marker prediction, but if it's 0, it doesn't line up at all with what happened. If you get an accuracy of 1, it means it lines up perfectly with what happened. What we're getting is actually somewhere in between that. So this is an example with a correlation of about 0.5. On the x-axis there, that's our prediction of what happened. On our y-axis, that's what actually happened. This is actually for Rust scores, and I'll come to this later, but this is a correlation of 0.5. So similar sort of information to what we're getting for fibre. And what you see here is what we're predicting is certainly not perfectly correlated with what we actually get, but it's giving us some information. For example, we can say the really poor clones, we can cut them out of the breeding program based on their DNA marker information, because they're never going to perform really well. Of the ones we predict to do really well, which might be up here, for example, most of them are going to do really well, but there will be some that only do moderately well. So it gives us some reasonable information for the breeding program, but certainly not perfect. And in the last part of the talk, I'll start to talk about how we might get to even higher accuracies of prediction. This is an accuracy of prediction of 0.8, where you really can focus in on those few clones that are going to perform really well. So I'll come back to the implications for the breeding program and how this can accelerate varietal development. But before I go into that, I want to go a little bit further in what we can do for predictions in sugarcane. And we can take our predictions in sugarcane a bit further than we can in other crops. And that's because sugarcane's a bit unusual in that it's clonally propagated. So you can, because of the way sugarcane's propagated, you're effectively getting the same genome just being planted over and over and over again in the fields. So it's exactly the same genome being planted again over and over again. And that's not the same as wheat, where it can be different and so on. So if we can come up with an ideal genome that we plant over and over again with our predictions, we can really take advantage of that. And one way we can do that is predict not just the effect of the genes that gets passed on generation after generation, which is the additive component, which is what people usually do in genomic predictions in livestock and other crops. We can also look at capturing things like how the genes actually interact, how pairs of genes go together to improve yield or improve commercial cane sugar. And if we can find a genome that captures that and benefits from the way those genes interact, we can capture that in our ideal clone and have a variety that captures that and gets planted out. So it can accelerate varietal development. So this ability to predict clonal performance is something a bit unique to sugarcane. and other clonally propagated crops like cassava. So we thought we should take advantage of this. So a researcher who was a student at the time of mine, Seema Yadev, looked at using this possibility in sugarcane, and in particular, how much could we increase yield by taking advantage of this opportunity. So the first thing Seema did was to look at how much of the variation between the clones that we had in our data set, how much of that was just driven by additive gene action that gets inherited generation after generation, and how much was driven by how pairs of genes interact to influence traits like total cane harvested, TCH, which is yield, commercial cane sugar, and fibre. And these are Seema's results. So if you just look at this bar that's labelled ADH, this is the proportion of the total variation between the clones, the amount of variation in the yield, that's driven by, in the red there, the additive bit, the bit that's inherited generation after generation, and the green bit is the bit that's affected by how the genes interact. And you can see in that bar, there's actually a large part of yield that is driven by the way genes interact. So a large part of the differences between how the clones are performing is driven by the interaction between the genes. The story is there's a bit less for commercial cane sugar. If you look at this bar labelled ADH, and even less for fibre. Sorry, the purple part is the additive component. The green part is how the genes interact, and the red is just noise, environmental noise. So just to summarise that, for yield in particular, a large part of how a clone performs is driven by these interaction of the genes. And because we can predict for a clone how much benefit it's going to get from the interaction of the genes, we can start to predict which clones are going to perform really well because they've captured how the genes interact well. And it turns out, Seema demonstrated, we can improve our accuracy of clonal performance for yield by up to 25%. So that's quite a substantial increase. For commercial cane sugar, the increase is 5%, and for fibre, there's not much improvement at all. But at least for yield, for variety development, we can really start to, just based on the DNA of a seedling, predict how well it's going to perform as a clone. We can do that substantially more accurately if we take into account this effect of how the genes interact. So that's good news for the breeding program. In her research, Seema went even further than that, and she looked at, can you pick the crosses, so which parents to cross that really maximise that variety performance. So maximise the performance of their offspring because they line up lots of favourable pairs of interactions of the genes to really boost the yield. So she ran some programs in the computer to look what would happen if we selected out specific crosses that really stacked up those favourable gene by gene combinations to boost yield. And she saw that you could get up to 30% increase in performance in total cane harvested if you designed your crosses to really stack up those favourable gene by gene interactions. So this made allocation, this design of crosses right back at the parental crossing stage, is something that should be really considered in the breeding program to speed up the development of varieties that stack together these favourable combinations to improve yield in particular. And that started to be considered in the breeding program. Okay, so up to this point, we were using methods that had been used for genomic prediction in other species. Predicting clonal performance was something that had been tried in dairy cattle, not particularly successful. Sugarcane, a lot more successful, because there seems to be a lot more gene by gene interactions that are important for traits like yield. But about midway through this project, artificial intelligence was getting more and more press. People were getting more and more excited about this technology, particularly in the media through things like ChatGPT and so on. And we thought, oh, we should try this for our genomic predictions in sugarcane. I have to say, artificial intelligence, more precisely machine learning, has a longer history than this in agriculture and in genomic prediction. We've actually been trying it for about 10 years, but as you all know, about 2 or 3 years ago, it became really the flavour of the month, I guess, and we thought we should really start to investigate seriously, does this help improve our predictions in sugarcane? Now, reasons why artificial intelligence, or more precisely machine learning, might be useful for sugarcane genomic predictions. This really relates to that complexity of the genome that I talked about before. So in other species where you have one copy of your chromosome inherited from one from mum, one from dad, we actually have pretty good biological models on how that might drive performance for different traits, and we incorporate that into our genomic predictions. Those models might not be as useful in sugarcane, where you have anything from 8 to 12 chromosomes. You may have 2 or 3 of those chromosomes switched on and actually working. The other chromosomes, the genes aren't actually functioning, they're not actually switched on. We have some information around that, but not a huge amount of information. So that's the kind of complexity that people have designed artificial intelligence and machine learning algorithms to deal with, where there's lots of interactions and the actual mechanism to get from, in this case, the genes to the trait performance, like the yield performance, is a little bit difficult to describe. So we thought it might be interesting to try these artificial intelligence, machine learning approaches to sugarcane. So I've been really lucky again to have a very bright student, Chen Song Chen, who has applied these methods to sugarcane genomic prediction. He's taken the same reference population that I described before, those 3,700 clones from the final assessment trials, and he's essentially run the same analysis, well, the same approach, where he takes all the clones from 2013, 2016, trains these machine learning models, and then tries to predict 2017. And these are some of his results for yield, TCH, commercial cane sugar, and fibre. So these again are the accuracies, the correlation of what we predict just from the DNA markers with what we actually observe. And there's lots of different methods here, so I won't go into any detail there. Just to say the The Brownie Gold methods are what we tried before. They're Seamer's methods, taking into account the additive and the gene-by-gene interactions. The purple is a machine learning method called random forest, and the green are other machine learning methods. The interesting thing is our existing methods actually outperform the machine learning methods still. The random forest, which is a particular type of machine learning method, it almost gets there. It's almost as accurate, but not quite. So it says a couple of things. One is our existing methods for genomic prediction are actually quite good. And in a way, you'd expect them to be because we've been running them for 20 years or so on and refining them. The machine learning methods, well, one reason they might not be performing as well is they actually need huge data sets to train on. If you think about something like ChatGPT, it trains on the entire internet. And here we're just training on 4,000 clones. So maybe over time, our predictions will get a bit better as we have larger data sets. But at the moment, what we're already doing is doing quite well. Okay, so I've focused on 3 traits to date, which is yield, commercial cane sugar, and fibre. About halfway through the project, again, we were lucky enough to get 2 more data sets which looked at disease and how susceptible nut clones were to 2 diseases, smut and pachymetra or root rot. And these were trials run by SRA, quite large trials. There was 4,900 clones for smut and nearly 2,000 clones for pachymetra. So these trials, each clone was scored for how severe an infection it had from 1 to 9. And it was also genotyped for those SNP arrays I described before. So 50,000 markers across the genome. So we used pretty much the same idea. We used these clones as a reference population to train the models to work out what each DNA marker was doing. And then we predicted a validation set. We kept part of the data aside as a validation set. We calculated how resistant those clones in the validation should be based on their DNA alone. And then we correlated it with how disease resistant they actually were to get an accuracy. My student Chen Song was doing this, and because his particular interest is machine learning, he included in his assessment of different genomic prediction methodologies, he included attention networks. And the reason attention networks are interesting is that's the very latest and greatest in machine learning. They're actually the methods that underpin ChatGPT-4 and other large language models and so on that people are using, enormous numbers of people are using for other applications. So he thought it would be interesting to apply these very latest methods to prediction of these diseases. So the good news is we can predict how resistant to a disease a clone will be from DNA extracted from a little seedling. We can predict that pretty well. The accuracy is moderate. The interesting thing is our existing methods perform a little bit better than the attention networks, so the current best machine learning approaches. So these 2 methods, they're called Bayes-C and Bayes-R, they're basically our best existing methods, and they're giving us the highest accuracies. The attention networks, machine learning, doing reasonably well, but not quite as well as our best methods. That's for smut. The story is the same for Pachymetra. We're not quite as accurate there. That's because we have less clones in our training set, in our reference set, but still quite high accuracies. So for both SMUT and Pachymetra, we're achieving these moderate accuracies. So with these moderate accuracies, I showed you before that plot of what a moderate accuracy looked like. I think these predictions of disease resistance will be very useful to just get rid of a whole lot of clones very early in the breeding program that are going to be susceptible. So if the DNA markers predict that they're going to be susceptible to disease, you just wipe them out from the breeding program and don't consider them anymore, and just focus on those that are going to be more resistant to take forward. So for disease traits, that's pretty clear how you use those predictions. So I'm going to finish up with just two more things. One is our ultimate test of genomic prediction, and then finally just how genomic predictions can be used in the breeding program to accelerate new varieties. So the last thing we did in the project was let me say, a slightly nerve-wracking experiment to demonstrate that genomic selection, genomic prediction really worked or didn't work. And this was our validation trial, for which we got the results in 2023. So this was if you like, a definitive demonstration that this really works. So SRA, we ran this trial, well, SRA really ran this, and the population that was used here, or the clones that we used here, were from a cross of two really high-performing clones that are used pretty widely by sugarcane growers. So they were crossed, a whole lot of progeny put out. They were screened with, in this case, 26,000 genome-wide markers. From that, we made predictions at the seedling stage, which clones would perform really well. So just based on the DNA markers, which clones did we predict would perform really well for total cane harvested, commercial cane sugar and fibre. And then together with SRA, we selected the best 90 clones, the clones that were going to be the best, and the 90 clones that were going to be the worst out of all those 500 seedlings. We made the selection actually on the index that SRA uses to weight total cane harvested, commercial cane sugar and fibre in its index for putting a relative emphasis on those traits. And I should say commercial cane sugar actually gets quite an emphasis in that index for selecting the best clones. So these top 90 and bottom 90 were taken out from the seedling stage and then grown up, planted out in a final assessment trial, and the harvesting was in July 2023. So if you imagine what we're going to do next, this trial's grown up, harvested. We're going to compare the total cane harvested, commercial cane sugar and fibre for those 90 best clones and the 90 worst clones. And remember, the top 90 and bottom 90 are chosen on an index of those three traits, and it really weights commercial cane sugar probably the heaviest, but there's a weighting on all three traits. And this is what happened. So these numbers, the average between the 90 bottom and top is 0. And the bottom 90 clones, if we look at commercial cane sugar first, that's where most of the weighting was. The bottom had an average of -0.3. The top had an average of 0.19. So the difference there is 0.49. And that's a highly significant difference between the top and bottom predicted, the top and bottom clones that we predicted. Total cane harvested, the difference wasn't significant. There's actually less weight on this trait in the index, but there was still quite a difference between the bottom and the top in terms of tonnes of cane per hectare. And fibre, again, there's actually a negative weight on fibre in the index. So the top clones had significantly less fibre than the bottom clones. And overall, if you look at the index value, there's a substantial difference between what we said would be the top clones and the bottom clones there. And that difference is quite significant. So with some relief, I can say that our validation trial demonstrated that our genomic predictions really were able to pick out the clones that performed really well and the clones that would perform poorly. So this validation trial really demonstrated to us that genomic selection was working well. So given that, we come to how is the breeding program going to change now that genomic selection has been validated? What's the best way to use genomic selection in the sugarcane breeding program? So just to remind you, this is what the current program looks like, where you've got crossing, progeny assessment trials, clonal assessment trials, final assessment trials, and release of new varieties, which is a 10-year cycle. So with genomic selection, what you can contemplate is crossing parents, picking out the best parents with your genomic selection. So it might be the best new varieties and so on, crossing them, creating a whole group of seedlings, screening those seedlings with the markers, predicting which ones are going to be the best clones, and then you wouldn't want to release those straight to the industry. We don't quite have the confidence in the technology to do that yet. That does, in dairy cattle, where I've done a lot of work, that does actually happen in dairy cattle now. Dairy bulls are released straight to the industry based on their genomic performance. In sugarcane, we're not quite at that level of accuracy, so you'd still want to grow out the best predicted clones in a final assessment trial, and then from that trial, release your varieties. This still speeds up that selection cycle, maybe cuts it down to 5 years instead of the 10 or 12 years. And you could also contemplate doing rounds of crossing before you go to a final assessment trial. So that would cut the generation interval, cut the cycle time down by half. The other possibility, a slightly more conservative approach, is you might just skip out, say, the progeny assessment trial step and go to a clonal assessment trial with your best predicted clones, and then get some information from those trials and take them through to a final assessment trial. We've actually modelled in some detail, actually, what this looks like in a computer simulation, where we simulated the current program in some detail and what these genomic selection alternatives, where you cut down the cycle time by about half, what that means for genetic gain, how quickly you can increase yield over time. So that's what this graph shows. That's a result of these computer simulations. In these simulations, you're starting to implement your genomic selection in at B5. And what you can see is the rate of gain is approximately doubling. So that means new varieties are released much more quickly after 5 years instead of 10 years, for example, with the same level of improvement. So you get the the same increases much earlier. So to summarise, based on the work we've done, the accuracies of genomic prediction we're achieving, our detailed computer simulations based on that, and the sugarcane breeding program, we should see a doubling of the rate of gain in the sugarcane breeding programs, which will accelerate the development of new varieties. We're going to get even faster discovery of those new varieties from this prediction of clonal performance. Because in sugarcane, a trait like yield, quite a bit of the yield performance comes from how the genes interact, and we can do a fairly good job of predicting that now. We're going to see an even faster identification of those varieties that have stacks of those good combinations of genes, which will lead to faster variety development. Machine learning is an interesting space. It's leading to lots of advances in many areas, as I'm sure you're aware, one of which is drones flying over sugarcane crops to find out traits like drought tolerance, nitrogen use efficiency, and so on. Fantastic for that. For clonal performance, it's not really helping us at the moment. It's almost competitive with what we do, but more work required there. As I said at the start of the talk, it's a very good time to be giving this seminar because we've just got the results from this validation trial, and basically genomic selection in sugarcane is validated. So we're pretty confident it's going to lead to have quite an impact on the breeding program. The future, what does that look like? Basically, the more accurately we can predict what a clone's going to do as a seedling in the breeding program, the faster we can go in releasing new varieties. And the way of doing that, one way of doing it is just to increase your reference set. So to have more clones with really good yield, commercial cane sugar, disease resistance information that go into our data set and have the DNA information there. So we have plans to work with SRA on this. Another area for improvement is to really start to dive into this complexity of the sugarcane genome. At the moment, we've made some fairly large simplifications in how we use the DNA information. But with new technologies coming out all the time, we'll be able to get a handle on what the sugarcane genome of different clones actually really looks like and use that in our predictions. We're also working together with SRA on things like stability of ratoon crops. So you can select varieties that have good performance across ratoons and don't get decreased yield across ratoons. So very finally, a huge thanks to all the people that have worked hard. This is 5-6 years of hard work by a lot of people. SRA team's been just terrific, right from sampling the final assessment trials to helping with methodology and analysis and so on. My team at UQ, the students, postdocs that worked on this. A really big thank you to the Queensland Government, Department of Ag and Fisheries for funding this work, and of course SRA for funding as well. And with that, I'll stop and ask if there's any questions.
Thank you, Ben, for this great presentation. So now it's time for the question, as you said. So to ask a question, you can either type it in the chat or raise your hand using the hand button located at the bar on the top of the screen. So unmute, and then when you finish asking your question, you can mute your microphone again. Thank you.
I work in forestry, and there's some nervousness about how well genomic selection can predict across generations. I was very comforted to hear about your validation experiment where you had a set of 500 progeny in a new generation and you were able to separate the bad from the good, basically. We haven't really got to that point in forestry yet where we can definitively prove that it can predict across generations because, as you know, recombination can upset the linkages between markers and causal mutations. And there's maybe some key points as to how well we can predict across generations, one being the size of the training population. Is there a sweet spot in how big that training population should be, like between 10,000, 20,000 individuals? And secondly, the design of the assay, should it be very dense and right across the whole genome? Just want to get your comments on those two factors influencing the success of genomic selection.
Yeah, great question, Richard, and good to have a question from you. I think there's a few things to consider here. One is, I think you need, in the order of what we have, which is about 4,000 clones, 4,000 individuals, before genomic selection starts to work well and robustly. We've seen that in different livestock species and in different crop species as well. In crops or plants, and you don't actually see the same thing so much in animals, but in crops, there's a lot of year by variety or year by clone effects that go on. So I think it's really important to have multiple year trials across multiple years that you're predicting from. I think you can get into a bit of trouble if you just have a reference population that's all within 1 year. And the classic example is that year was a really wet year. So you're actually pulling out the clones that do well with a lot of water. But when you predict into a year that's a bit drier, you run into trouble. So having good multi-year trials is quite important. And then I think, having a serious marker panel that really does go across the genome is quite important. In the early days of genomic selection, people were using densities like a couple of hundred markers, but I think something like the 20,000 that we routinely use in sugarcane, or up to 50,000, that's where you should start, basically. Having said that, in really inbred species like wheat, we do get away with about 5,000 markers because the genetic diversity is really low. But if you've got a fair degree of genetic diversity, 20,000 is a good starting point.
Yeah, I think in forestry species that are recently domesticated, linkage disequilibrium.
It's going to be huge. Yep.
Yeah.
You might actually need more like 100,000 markers.
Well, that's what I'm thinking. Yes. Yeah.
Yep. But having said that, I think in now in 2024, that's not the main limitation. The main limitation is actually getting good trait data in a good trials, multi-year trials.
Yeah. In the early days, the molecular geneticists would say, well, you don't need to phenotype anymore, but
no, I think it's the reverse.
Yes, you need to constantly phenotype.
Yeah, just on that, the genomic selection gets you a couple of generations ahead, but
you're very confident you can predict across generations.
A couple of generations ahead. I wouldn't go as far as saying we can predict 5 or 10 generations ahead, because I think then, as you pointed out, recombination gets you and starts to break down your marker mutation, the real mutations that are affecting your traits, starts to break down those combinations. So you always need some phenotyping coming along behind your breeding generations.
Yeah.
We've seen that very clearly in cattle, but you get maybe 3 or 4 generations ahead. Yeah.
All right, thanks. Karen Atkin, do you want to ask a question?
Hi, Ben. That was a great
Okay.
talk. Thanks very much.
Thank you.
I noticed that your validation population was a cross between two parents, so all the progeny were half sibs. So that kind of reduces the complexity a bit. Can you comment about that?
Well, that's very interesting because I had a mild panic attack when I saw that that was going to be the validation. Apologies to my SRA colleagues. I can see why the validation trial was set up like that, because they're two very interesting clones. But there's been quite a lot of commentary about genomic selection that all it does is pick between families, picks the good families or the bad families, and doesn't really give you the ability to predict within a family. So if you look at the genomic selection literature, some people claim that. But because of our validation trial was strictly within family, it was one cross, as you say, that's actually a much harder thing to do, to pick out the good clones within a family, because you've got to pick up the within family segregation well. And we managed to show that we can do that quite well. So I'm actually pleased in a way, given we got a good result, that was how the validation trial was done.
Yeah, I guess in sugarcane, because there is so much variation because of the ploidy level, you do get that wider.
Yeah.
You know, it probably would be easier to do it in a cross. But having done this, do you think that the breeding program should change to having larger populations from particular crosses?
Yes, I do. I think what we should do is predict which, exactly like SEMA did, which crosses have the potential to produce really good varieties, and then basically have lots of throws of the dice on those families so that you get those stacks of chromosome chunks from that cross that give you the really good predicted clonal performance. So you could imagine a breeding program where you run it a little bit like it is now with set family sizes, but the crosses that are predicted to have really good clonal performance, you just throw the dice many more times on those, so double the family size or something, so that you actually get progeny that have those stacks of chromosome chunks that you predicted would be really good. So, yep, that's a good thing to do, I think.
Yeah, that sounds good. Okay, thank you.
There's a question in the Q&A. So should we be cautious in getting variety that are all resistant to a single disease and increasing the strength of a disease? Should we have varieties that aren't as resistant? That's from Bob.
Yep, that's a good point, and I think I see where that question came from. And I think what we should actually do is make sure we preserve some genetic diversity in the breeding program, so that there are bits of DNA floating around that are potentially resistant to future diseases that we haven't seen yet. I think if we, as the questioner I think is suggesting, go very hard for resistance to one disease and just, for example, extreme is make all the future breeding program based on one or two clones that are resistant, then we're going to get into trouble with other diseases. And the classic for this is banana breeding programs, where everything is Cavendish, and obviously Cavendish is highly susceptible to some diseases. So we should make an effort to preserve genetic diversity, and we haven't done it. We've done it a little bit in sugarcane, but certainly in our wheat breeding programs, that's becoming a major focus. So there are good ways of doing that, and we should start thinking about that. And incidentally, the genomic information, the marker information, that's great information for preserving diversity, because you get a snapshot of what the genome of each clone looks like. So you can start to see when your diversity is really narrowing down. So, yep, good point.
We don't want to narrow the genetic base that much that.
No. It's a real risk. So you have to balance that
You have to balance
along with spinning out these really good varieties. So it's a balancing act. Yep.
Yeah. I'll just get some a question from Mohammed Karam. Hi, Ben. Great presentation. Would there be a possibility of screening with 50K or 100K as current screening is with the 26K genome SNP marker? Is it applicable to sugarcane genome?
The array we're using is Karen's array, and I can tell you it works very well in this material. It's about a 70K array, 70K SNPs on it. We cut it down to 26K in our routine evaluations because we know those 26,000 work extremely well. They're polymorphic, they have 2 alleles in the material, and we just use them for a routine set. You could go back and I think extract more SNPs off that array that work well in other germplasm as well. But certainly for the clones that SRA uses, it works well. I can't really speak for clones outside the breeding program. Maybe Karen would be a good person to talk to about that.
Shemsa. Hi, Ben. In terms of resistance screening for Pachymetra smut, do you think there will be a variation, that is, different outcome if we use different population?
Different population, yep. It really depends on the diversity in that population and how good the phenotypes are. I mean, the phenotypes or the trait records we have from those final assessment trials, they're pretty good. They're pretty good trial designs, good replication, and so on. If you have trial data that is less well structured and so on, you might get lower accuracies, but it really depends on how good your trial information is. Another thing to say is if you have very diverse material, like really diverse, more diverse than in the SRA collection, you're probably going to have lower accuracies there as well, because for the way to think about it is for each segment of the genome that's in the population, you're probably going to have less trait information on those segments of genome. And that leads to lower genomic prediction accuracies. So one thing we see clearly across species is the more diverse they are, genetically diverse, the bigger the reference set needs to be to get good predictions. The flip side of that is the less diverse they are, the smaller the reference sets are. So in wheat, where there's less diversity, we actually need quite small reference sets to get pretty good predictions. But in something like beef cattle that are really diverse, lots of different breeds and stuff, we need enormous reference populations, tens of thousands of individuals with traits.
Anthony Young, nice presentation, Ben. There has been significant flu in sugarcane varieties through the years, with hundreds released in the past 100 years. Do you know of any analysis that shows whether it is a loss of productivity in older varieties or whether the replacement is simply driven by genetic gains of new varieties?
So people in the SRA program are probably better place to answer that, but there has been the driver is really increased genetic gain for the improvement in those varieties. And there's some studies that have gone back and documented the improvement from those varieties in terms of traits like yield. And you can see approximately 1% improvement in yield per annum. But of course, that accumulates over time. So across 20 years, it might be 20%. It has been, I do know that it has been harder and harder to squeeze out that 1%. And we hope things like genomic selection and predicting clonal performance, taking into account how genes interact and so on, will drive up that improvement again.
Jason said that maybe it is worth mentioning the current cost of genotyping, which is one of the reasons it is only being used as a tactical small scale.
Yes, that's a very interesting point. So from genomic selection, we can demonstrate you double the rate of genetic gain. To actually implement that, you need to screen, I think we worked out 6,000 clones every year, seedlings. And the cost of current genotyping is, depends who you bargain with, but perhaps between $170. So you can sort of do the sums in your head. That's a big amount of money. In cattle, where we now genotype hundreds of thousands of animals every year, the cost is down around $30 per individual. And there's talk of new technologies that go down to $15. So I think in the future, we're going to be able to get it down a lot further, combination of big volumes and new technologies, but we're working towards that. And when we can do that, that's when genomic selection in sugarcane will really take off, when we get it down to $30, $25. Yeah. Good point, Jason.
Thanks, Ben. Armin Wessel. Once you have the current breeding stock validated with genomics, you can select the best candidates in that pool. How do you integrate new breeding materials from overseas programs? Can the genomic testing, if they do it, be helpful, or do you have to wait until new genomic information is confirmed with evaluation of offsprings?
Yeah, that's a really good question, and we're face that challenge in a number of plant breeding programs we work with. So what we're actually doing in those other plant breeding programs is diving a bit deeper into the genome to work out which chunks of genome are really giving those lines or clones from overseas or wherever it is, the properties that we want. So it might be disease resistance. So we would try and work with our international partners to run some genomic analysis, identify which chunk of genome is really driving resistance to a particular disease. And then when we pull that clone into the breeding program, we can use the genomic information. You have to make the crosses between the parents and that new clone, but we can really use that to work out which of the progeny have that favourable chunk of genome and discard the rest, and then focus on going forward. You in fact want to go a bit further than that, because if that chunk of genome is also associated with a really big yield penalty, you may not actually want to use it. The clone may look really good for disease resistance, but it may have a really big yield penalty. So some information on how that chunk of genome actually performs through doing a trial with the progeny that contains it, I think is going to be important. I don't think you ever really get away from having to do some trait recording, particularly before you release a new variety. Yep, but I think the upshot is the genomic tools help you focus in on what bit of genome is useful to pull into your breeding program.
One asking the cost analysis of conventional selection versus genomic selection.
Yes, it's an interesting question. With the genomic selection methods we're proposing, there's an increasing cost due to all the genotyping of the small seedlings you have. There's also a decreasing cost because you don't have to do some of the intermediate stage trials. So there's a trade-off there. But at the moment, because of what Jason referred to, the cost of genotyping is a bit high. So that has to be offset by the increased genetic gain you get. But in the future, and what we've seen in other species, is it can be sort of cost neutral because you have less trials to run, but the money you save from that you have to put into genotyping. But the overall benefit is increased genetic gain, more frequent release of better varieties.
Clive Gannon, did you include disease resistance in your top-bottom assessment, or knowing that there is some predictability in these traits?
No, that wasn't included in the validation trial. So that would be very good to include in the future. And we need a way of combining that nicely. So if you have a seedling and you do genomic predictions, you'll have disease information, yield information. How do you weight that relative information? So that's work still to be done.
Richard had another question. Did you use imputation in the research undertaken with SRA? A possible low-cost assay platform is YGS at low red depth. This combined with imputation to recover missing or poor genotype call data is an attractive option. What are your thoughts?
Yep. So in this particular example, we didn't. We just used the SNPs on the array. And those, the 26,000 SNP we used, the call rates were 95% or higher. So we didn't do much imputation. We did a little bit of imputation to fill in the missings. But what you're describing, the imputation from low coverage sequence data, is a really good idea. And if you're interested, we've just published a paper in cattle on how that can be used cost effectively. And that's using nanopore sequencing, these portable sequencing devices. So I can send you that if you like. It works extremely well. And that's one way we think we'll get down to $20 for genotyping. I have to say, though, in sugarcane, there's some challenges because low fold coverage imputation works well when you've got a simple genome, a copy from mum, copy from dad. I'm not sure it's going to work so well if you've got 8 or 12 copies of the genome. That's actually a lot, if you do low fold coverage, a lot that you miss. So we have to think about how those kind of technologies can be targeted to sugarcane. And people are doing that, yep. But it's, again, more complex in sugarcane.
Greg had a question. He has his hand up. Do you want to go ahead, Greg?
Okay, is it going to work? Is it going to work? Here's the question.
It's working.
It is working.
Yep.
Okay, thanks, Ben. From what I understood, you used 3 years of trial data where you'd had genotypes and phenotypes to then predict for the 4th year of testing. Is that what I understood you did?
Actually, 4 years and we predict the 5th year, yep.
Okay. So what was your control method without using the genomic information? So did you use, just use the phenotypic data from that day, those 4 years to predict the performance in the 5th year? And we're able to show an improvement of predictive ability by adding the genomic information.
So what we're predicting is from the way to work in genomic selection, predicting from the seedling how it performs as a clone. So you don't have, at that point of prediction, you don't actually have any phenotypic information. What you would have is pedigree information, who's mum and who's dad in the cross. And we haven't done it, but the people at SRA have predicted from the pedigree what that clone would do when it's as a seedling, when it grows out to be a clone in a trial. And
sure, that's what you do in the breeding program, but in the validation you carried out, you weren't doing that. You were just looking at the same sets of families, the same sets of clones tested in 5 years. And then you said, going from 4 years, predicting the 5th, you got this predictive accuracy. But you only did that by doing the genomic model. Did you just use, a phenotypic only data?
So in our validation trial, I'm talking about strictly the validation trial at the end.
Yeah, no, I'm not talking about that. That was very encouraging, I agree. But in the previous step, you showed a predictive accuracy, 0.4, 0.3, but only with the genomic model. What if you just used a phenotypic data without the genomic information? So you've got to be able to show there's an increase in accuracy applying the genomic information, I would have thought. But you don't have when, if you I agree. In the routine breeding program, you don't have it, but in the validation you carried out, you did have it.
So if you correlate the final assessment trial data with the final assessment trial data, it gives you an accuracy of 1 because you are correlating the phenotype with the phenotype.
No, if you use the data from the previous 4 years, you have already performance data on those same genotypes, same clones that were tested in year 5. So you can use that data.
Okay, I get where you're coming from now.
Yeah.
What we did to make that prediction quite rigorous was we took our validation set, so the clones in 2017, and we dropped out any clones from the previous 4 years that were the same as those clones in 2017. So they're a completely new set of clones that you've never seen before. Sorry, I didn't describe that. You're quite right.
Okay, that sounds much more encouraging. Thanks.
Yep. So I guess the point is, good pickup, but they're clones that you've never seen before. So how well do we predict clones that you've never seen in the field before? Yep, good pickup. Thanks for that. Sorry, it took me a little while to get that.
All right. Well, we'll have to end a session today because we're running out of time. Thank you again so much, Ben, for presenting today. I think we can all agree that it was very informative. And thank you all for your attendance today too. I hope you have enjoyed today's webinar and that you will join us for the next webinar on the 21st of February. And for information on this webinar, you can please visit our events page at the SRA website. So thank you all very, very much. Have a good end of day and a great weekend.
Okay, thank you all very much. Great questions. Really appreciate that. Thank you.